Our brain is a very complex organ. It is being studied by a lot of professionals like neurosurgeons, neurologists, neuroscientists, psychiatrists, and psychologists. So also, I have to say that biomedical engineers also study the brain, and we use a different approach to study the brain. And this project is the project that I've done. A major part of my project that I've done here at the Medical College of Wisconsin in the last 3 years, and it's a collaborative effort between me as a biomedical engineer and the neurosurgeons and neurologists in Weill Cornell Medical College. And I want to show that how biomedical engineers can use our engineering tools to provide insight to some neurological or brain problems that cannot be attained by the traditional approach of neurosurgeons, neurologists, and some other medical professionals. So the technique I'm going to talk about today is called deep brain stimulation. So this is a deep brain stimulation system in the human body. It has a electrode, has multiple contact inserted deep inside the brain. It delivers a constant pulses, either current pulse or voltage pulse, to the brain structures that are responsible for some neurological diseases. And this electrode is connected through a wire under the skin all the way to the upper chest where the implanted stimulator is placed. The implanted stimulator is generated pulses, and the battery is also inside the stimulator. And deep brain stimulation is an established therapy for some neurological disease, especially movement disorders. And this video shows the dramatic effect of deep brain stimulation in some patients with movement disorders, such as Parkinson's disease. So this older gentleman is Parkinson's patient, and this the left is when DBS is off, and the right is when DBS is on and played against rather quick. So you can see the dramatic difference of his walking gesture after the DBS is turned on. So this is not a patient who has the essential tremor. When the DBS is on, he can hold his hand still on his leg. And when DBS turned off, he started to shake involuntarily. And this apparently affects his daily activities. He cannot even drink water from a water cup. So when the DBS is on, his tremor disappeared almost instantly. So these two videos just shows the dramatic difference DBS can produce in patients with movement disorders. With our brain have different part of different functionalities. So maybe it gives us hope that if we stimulate different part of the brain, maybe we can cure some other diseases using the deep brain stimulation. In fact, deep brain stimulation has been approved by FDA for a number of neurological disorders, such as essential tremor, Parkinson's disease, dystonia, and obsessive compulsive disorder. And it's also been actively studied to treat depression, epilepsy, chronic pain, and disorders of consciousness. So disorders of consciousness is something I'm going to talk about today. So let's first talk about what is consciousness. Consciousness, in my opinion, consciousness is the biggest question in the universe because our understanding of the universe comes from our consciousness. So consciousness is like our inner universe to generate our self-awareness and to understand, to observe the outside from the neurological point of view, consciousness is maintained by a certain level of arousal. Arousal means our readiness to engage when there's a stimulus presented in the environment. For example, if you're doing nothing in a room, so you're awake, you're bored, you're doing nothing, but you are conscious because you're ready to engage. For example, if your cell phone rings, you would immediately respond by picking up the phone and answer it. So arousal is maintained by this brain network. It involves with a lot of brain regions. The brain receives all the sensory input from every part of our brain, and it converges into the midbrain, called reticular activating system. And then there's a central thalamus. Is sort of like a hub that convert the signals to a wide range of cortex regions. So there's a widespread projection from the central thalamus to the cortex to maintain a certain level of arousal when we're awake. So what are different kinds of disorders of consciousness? So this spectrum shows the different level of our consciousness state, from all the way from the brain death to normal. So on the leftmost side is the brain death. So right above it, there's a state called coma or vegetative state. Coma is more like acute state. When people had an injury, initially he was in coma, and then he survived in the first couple of weeks. He would progress to vegetative state, or even better, to minimum conscious state. So for vegetative state, the patient has total cognitive loss. He has no behavior response to the environmental stimulus. In minimum conscious state, we call MCS. In minimum conscious state, the patient has some residual cognitive functions, such as he can fixate and he can trace in a moving object in a room. But most patients don't have the functional communication. Some patients have some functional communication by answer yes or no questions, by blinking their eyes. So I'm going to talk about some of the famous patients at different famous patients of disordered consciousness. And Terry Shiver is a lady who whose death caused a huge national debate whether it's legal and ethical to utilize somebody who apparently cannot make a decision to live or to die. And then there's another patient I want to mention today is Don Herbert. Don Herbert was a New York State firefighter. He got into minimum conscious state when he rescued a house fire. And when he was entered the building, the roof of the building collapsed on him, pressed the oxygen tube. So he was deprived of the oxygen for several minutes before he was rescued. So which caused permanent damage in his brain. He was in minimum conscious state for many years until 10 years after the injury. One day he started to talk and to function. So that's normally considered a miracle because late recovery from vegetative state or minimum conscious state is very rare, especially someone in that state for 10 years. So our questions here is maybe can we use our modern technology? Can we actually awake patients in vegetative state or minimum conscious state by electrical stimulation? So that's the big picture of this research. And so what causes disordered consciousness? Traumatic brain injury is one major cause of disordered consciousness. And this is as I mentioned earlier, this is the brain network that is involved with our consciousness or of our arousal level. And the side that is vulnerable to the injury is here. That is the midbrain and the thalamus. So in some patients, they have preserved widespread lead work from the central thalamus to the cortex. Those lead work are being preserved, but they're underactivated because the lower brain structure are injured. So they're recruitable, but they're not being recruited because they don't got any input from the lower structures. So that's why deep brain stimulation might be able to play a role in this kind of scenario. Maybe we can stimulate the central thalamus, so we can recruit the widespread neural network so that we can awake the patient. This is exactly what our collaborator, Nicholas Schiff, his idea. So he used the deep brain stimulation on a patient, 38 years old, 6 years after the injury, and after he applied the deep brain stimulation on this patient, so this patient started to talk and function. So that's really considered a miracle in this patient community, and because this wide received study by medical professionals and by the patient community and their caregivers and their families. And Dr. Schiff was named by Time Magazine 100 most influential people in the world in 2008. So, but before this study came, okay. So this paper was, so this is the MRI of this patient. You can see this patient's brain is highly deformed because of the injury. So they have to implant the electrode sort of in an asymmetrical kind of fashion to hit the target and to avoid certain structures. So this two trajectory, you can see that's the trajectory of the electrode implantation. And but before this, this technique can be applied to a bigger population to become a standard therapy to people who have disordered consciousness after traumatic brain injury. We have to answer some very important questions. These questions are why it works. So that we have to answer unveil the mechanism underlying deep brain stimulation in regulating arousal network. And we have to answer another question is where to stimulate, means stimulation targets, because central thalamus is not just one structure. There's a lot of substructures in the central thalamus. So which part of the central thalamus, which nucleus we should stimulate to achieve the optimal results, and how to stimulate, what means how to choose the stimulation parameters like waveform, what kind of waveform, what kind of frequencies you use, and amplitude to use. So in this study, I'm going to answer trying to answer the second question, where to stimulate. So let's look at the anatomy of the central thalamus. So central thalamus there's a lot of substructures in the central thalamus. So this is the thalamus. This part is the thalamus. So central thalamus means the center part of it. There's major structures that are responsible for the arousal network. It's this nucleus called CL, centrolateral, PF, whatever, I don't remember. So, and CM and CM, those are central thalamus. RET is reticular nucleus. It's not really part of the central thalamus. It's just the outside, like a shell of the thalamus, sort of the boundary between the thalamus and the rest of the brain. So our question is, and also there's fibers that are running around the central thalamus. They're also involved with the arousal network. So our question is, where should we stimulate to achieve to increase, to facilitate the arousal to the behavior response of the patient. So to answer this question, we collaborate with Dr. Schiff and some other scientists, neuroscientists in Weill Cornell Medical College, and we use the deep brain stimulation on monkeys. So this is the system we designed in the monkey brain. So this is two chambers here that to implant the stimulating electrode and the recording electrode. So this is the central thalamus, different structures we built. And so today I'm going to only talk about the stimulation part. So I'm not going to talk about the the recording part of this project, although that is a huge part of the project that will answer the mechanism of this therapy. But I'm going to talk about the stimulation part, which is this red chamber here, and the electrode in the central thalamus. So the first challenge we have is how to implant the electrode and to hit the target, because this target are really small structures. You can see this CL is a really thin structure in the central thalamus, and there's CEM, and PF. They're really small structures, and this is the atlas of the brain. Unfortunately, our brain is not atlas. Our brain is if you look at it in our MRI, this is what you get. So the thalamus is just this. It's just a bunch of gray matter. It does not have the boundaries between different nucleus. So how can we hit a target? And I have to mention that the surgery is done by our collaborator who are neurologists. So they're not really neurosurgeons who do monkeys a lot. And apparently we cannot afford a lot of mistakes because monkeys are very expensive. We spend months to train them before the surgery, so we cannot afford a lot of mistakes by misplaced electrodes. So I have to, as a biomedical engineers who specialize in image processing and visualization, how we can help them to plan a surgery. So this is what we did. First of all, we used this atlas, which is 2D atlas, a series of 2D slices, and we built 3D structures out of it. So this is the thalamus in 3D, and you can see, if you look inside, you can see the small structures. Those are different, CL, CEM, PF in 3D. And then we registered this atlas to our monkeys MRI. So we expressed all the structures in the monkey's brain, so become a subject's specific 3D structure, 3D atlas, and then we incorporate the chambers and electrode into this navigation system we created. So we basically can do a virtual surgery on the computer screen to determine the implant angle and depth of the DBS lead before we do actual surgery. So then so we can have the confidence that we are hitting the structures, really small structures we are trying to hit. So this is a surgery picture, and this is the monkey. You cannot see, but this is monkey's skull. And this is 2 chambers, and this is, they're trying to implant this electrode into the brain. So after the surgery, how can we be sure that we hit the target that we are trying to hit? So we use the post-operative electrode localizations, because the electrode and the chambers are a metal, so they have different intensity in the CT image. So we can do 3D reconstructions. We can get halos of the chambers and electrode, and then we match them with the 3D structures, and we can determine the final location of the electrode. And this is the final location of the electrodes. We have 2 electrode leads, each have 6 contacts, and this is central thalamus, CL, CMN, and PF. And so we are pretty much on target because of this CAL4 surgical planning in the computer screen, and then on the surgical room, and we pretty much hit the target we want to hit, especially the blue electrode. So he's pretty much right on target, because the electrode actually stimulates a volume. So it's actually the volume of activation actually impinging upon all the structures. So which is good. So this is a sort of 3D view. You can look at in 3D and how this 2 electrode impinging upon different structures. And if you want to pull some other structures, You can also see, this all the structures. This is reticular nucleus, and this, this is some neurons that have been activated. I will show you later. So basically, this 3D view is kind of a communication tool for us to communicate with the neurosurgeons and the neurologists. We can provide them with a 3D PDF, so they can open with their Acrobat reader, so they don't have to install any software on their computer to examine the structures and the simulation neurons being activated in any computer. So.
I have a quick question.
Okay.
What's the scale of this?
What's the scale of the electrode?
Yes. Or
Okay.
Like this.
Yeah, I can provide you the scale of the electrode. The electrode is 0.5 millimeter in length, so that's half a millimeter. So it's pretty big electrode because we are trying to sort of activate a volume.
Okay.
So, but then the oldest electrode are, they can turn on and turn off individually. So you can have a combination of this one is current source, this is current sink, and others are just passive electrode. So you can have a lot of combinations for electric configurations, how to deliver the charge into the tissue. Why this doesn't work now? How to go to the, okay. Okay, so after we implanted electrode into the monkey brain, so we started to we trained the monkey. We actually trained the monkey before the surgery as well. So we trained the monkey to do some tasks. It's called sustained attention task. So basically, the monkey is facing a computer screen. The computer has a dashboard here, which initially is red. So when the monkey fixates on the computer screen, after 1 second, this red dashboard started to flash. So it's a warning sign that, the monkey, tell the monkey that it's almost time to perform the task. After a delay period, variable delay period of 1 to 4 seconds, this red dashboard turns green, so indicate that it's the go signal for the monkey to perform the task. All monkey needs to do is to touch a bar and release it. And then if he does this in a certain time window, he gets some rewards, which is a drop of water, actually. So that's the task we train the monkey to do. And we test the monkey before the deep brain stimulation and after during the deep brain stimulation. And what we found is that the behavioral performance strongly depends on electrode configurations. What I mean electrode configuration is, as I mentioned just now to answer the question she raised, each electrode contact is individual channel that can be turned on, turned off individually. So we basically can have a lot of combinations. For example, in this configuration, these two electrodes are cathode, so they're current sink. So these two electrodes are anode, they're current source. So maybe this configuration is effective. It calls the behavior facilitation of the monkey. So we have done a lot of combinations of the electrode configurations, and we found that certain configurations cause behavioral suppression. For example, this is some configuration. This is the summary of the configurations that cause the behavioral suppression. You can see, and this green is when DBS is turned on, and the white is before pre-DBS. You can see that for some configuration, the behavioral score decreased after the DBS turned on. But for some configuration, there's literally no change. But then for some configuration, there's an increase of behavioral response after the DBS turned on. So this is interesting. So can we answer the question, our initial questions? Can we use this information to answer questions that where to stimulate? What is the stimulation target that we can achieve the optimal behavior facilitation or increase the arousal of the monkey. So that's our question. So that's the observation, or the neuroscientists they observed, and how biomedical engineers, how we can use our tools, computational tools, to provide them insight that cannot be obtained by the traditional approach of neurosurgeons, neuroscientists. So I don't know why this one is here. Sorry, this should be deleted. So this is what I did for the computational models, and for each electrode configuration, for example, for this one, so this is the cathode, this is the anode. So I use finite element model to simulate the voltage distribution in the tissue, and then I place neurons around axons around this region. So this is the two electrodes. Okay, so two electrodes actually is tilted in a certain angle. It's showing there, it's tilted in a certain angle. So I place neurons in a box here that each node is the location of the axon, that center of the axon location. So I place them in this box. And for each location, I oriented the axons in multiple directions. Actually, altogether, there's 13 directions. And X, Y, Z is the main axis direction. And then there's X, Y, Y, X, and Y, all these combinations, Y, Z, Z, Y, and there's 6 of them. And then there's a cubic diagonal direction, X, Y, Z. So basically, altogether, there's 13 directions. I oriented axons in each location so that the axon is sort of, the orientation cover the entire space. But what is the actual fiber orientation? We use the diffusion tensor image to figure it out. So this is a diffusion tensor image of the monkey, and so the color of this diffusion tensor image indicate the anisotropy of a certain brain region. If they're red, means that means this FA value, fractional anisotropy, is 1, means they're highly anisotropic, means the water molecule is like a line. Water molecule would really prefer a certain direction, but if they're 0, means there's no preferred direction for the water molecule to diffuse. So means there's no directional preference. So this is the diffusion tensors of this part of the region in the brain called internal capsule, which is highly anisotropic. You can see there, each tensor is apsoid, have a certain directions. You can see the fiber, will run around this direction, pretty much, upside down directions. And so we match this direction. For every node, we match the tensor direction with these certain directions to determine what is the actual axon directions we should simulate for every locations. And then we find the threshold of that axon to initiate action potential. And the results we get is for all the configurations our collaborator, the neuroscientists, they did, we generate a point cloud of neurons, the location of point cloud of neurons that are being activated by this configuration. And this minus means for this configuration it calls behavioral suppression. An equal sign means it doesn't cause behavioral change. And a plus sign means it calls the behavioral facilitation. So this basically gives us some kind of idea of the location dependence of the behavior response of the monkey. So the next step, what I did is I combine all this, I group all these configurations together. All these equal signs means the configuration doesn't cause the behavior change. the configurations that cause the facilitation, I group them together, and then I will find out the electrode location, the neuron locations that are being activated by configurations that cause suppression, no change of behavior response, and facilitation. So we can see a kind of behavioral, the kind of a location dependence from the 3 kind of point clouds. And the next step I did is how to quantify this. Is I count how many neurons in each structure. So each, for example, each neuron falls into one of the structures. So I count how many of them in each structure, and what I get is the percentage of neurons falls into all different structures, and we can see that for suppression, for CLPC neurons, it mostly causes no change or facilitation, but for CMN neuron, it causes strong suppression. So that we can get some quantitative measurements of this location dependence. So using this approach, and we conclude. So actually here I have another neuron called MD I didn't include earlier. So MD is a neuron here, so it's here. So MD is another neuron. I didn't put them together because otherwise it will block the electrode. You don't see the electrode. So anyway, so the conclusion we make from using this approach is behavior facilitation was most strongly correlated with stimulating the centrolateral, paracentral, and the medial dorsal nuclei. And behavior suppression can be produced by stimulating the central medium nucleus. So this is a conclusion we can make through our computational models and image processing and visualization that can only be achieved by biomedical engineers. And so I think this is another conclusion. Most important is, as a biomedical engineers, how we can collaborate with other scientists and how we can how we can answer questions that cannot be answered using traditional approach of other medical professionals to study the brain.